I'm really excited to share with you some of the recent work that we've been developing in our lab, and specifically a technology called DAF-seq. And really what this technology comes down to is trying to understand is how does the genetic blueprint within the cell work? And what we know is that this genetic blueprint, the cell can do many things, but one of the critical things that it does is actually tell the cells when and where to regulate and to express different genes. And specifically, this genetic blueprint or the DNA is stored in the cell in the form of chromatin, which what that is a bunch of proteins that interact with the DNA and tell the DNA sort of when and where different parts of the genome are going to be expressed. And this is mediated by different types of proteins, as is depicted in this diagram here. What we know is that one of the ways that somatic variants can actually impact the cell and impact the function of the cell is by altering how exactly these proteins are actually being formed within inside of the cell. And specifically, if you imagine this from a genetic perspective, you would have, say, a genetic variant that exists here, and maybe this is a somatic variant that exists there. And what that can actually be doing is now disrupting the ability of this protein right here to actually interact with the DNA. And that is the functional effect that we really want to be able to measure inside of the cell to understand what is the function of this catalog that we're developing as part of SMaHT. Really, the challenges that we face when trying to understand this is that we need to be able to accurately identify these rare somatic variants. And also, we need to be able to actually accurately identify what the function of those rare somatic variants at the exact same time. And specifically, how do they disrupt the chromatin or disrupt how these proteins are interacting with the genome? And furthermore, we need an ability to do this in a targeted manner as well as at individual cells, so we can do this robustly across the different variants that we're identifying as part of the SMaHT catalog. So our lab has been developing different tools and technologies for accomplishing this. One of the earlier tools that we developed was this tool called FiberSeq, which uses a non-specific adenine methyltransferases to selectively stencil where proteins are bound along the genome. You can almost imagine that these proteins or enzymes are being used as spray paint, where any spot along the genome which is not being physically bound by protein is actually being spray painted with this enzyme. And this enzyme, what it will do is that when it sees a piece of DNA, it will modify it in form of methylation. You can use long-read sequencing approaches to sequence these DNA molecules that have been stenciled or spray painted. And this enables you to directly identify both the genetic sequence of these molecules, as well as these prints in terms of stencils for where individual proteins are bound along the genome at these sites. We've been leveraging this approach across multiple different studies, and it's actually been quite robust and quite powerful. However, one of the major challenges when it comes to somatic variation is that these adenine methylation events or these stencils are not actually preserved upon DNA amplification, which means that it can be challenging if you're trying to do this at specific spots along the genome, or if you're trying to do this within single cells. And so really what this left us with is we have a methodology that can really accurately identify these somatic variants and determine their functional impact, but we're not able to get this last part of the challenge, which is doing this in a targeted or single cell manner. So how we addressed this challenge was really sort of redesigning how we're actually spray painting the cell when we're doing this initial stenciling step. And really what this means is we need a new spray paint to go after. To address this challenge, what we ended up actually doing was designing another spray paint, but this is actually a spray paint that will now modify the DNA in form of these cytidine to uridine alterations via a cytosine deaminase. And the nice thing about this particular type of spray paint is that these cytidine to uridine modifications are actually preserved in the DNA sequence upon amplification in the form of these C to T changes, enabling us to do this in a more targeted manner. This is just a diagram sort of depicting what exactly this sort of experimental paradigm looks like, but specifically similar to the last methodology I showed, we're treating cells with this enzyme, and then we'll amplify either the whole genome or a specific region along of interest, and then we'll actually get both the genetic sequence as well as the chromatin architecture, but now in the form of these deaminated cytidine events. And this, what I'm showing you here is actually some data from FiberSeq at a given locus. And what I'm showing you here is each individual gray bar that's going across here is a single molecule. So that's one of these single chromatin fibers that's depicted here, and showing you how it's actually organized in terms of, there's areas of accessibility which is sort of analogous to this regulatory element you're seeing right here, as well as areas that are closed down, which have these nucleosome patterns. You can appreciate that there is some differences between individual molecules in terms of how they are structured. But one of the benefits of the DAF-seq approach, unlike fiber-seq, is that say if we're only interested in this one area of the genome, say this area that's only around 4,000 base pairs in length, we can actually target that area and selectively amplify it and get really high coverage of how exactly this specific area is being regulated. And this is nice because this now enables us to go really deep in terms of identifying rare variances I'll go over in a couple of slides from now. Another big advantage of this is that, as you can see from this earlier sort of depiction I showed you, is that oftentimes these regulatory elements are bound by multiple proteins, which the co-occupancy of these multiple proteins is really essential for determining how the function of that regulatory element is going to exist within the cell. And when we actually look at the sequence in actually the architecture of these individual molecules within the promoter, what we can see is that we're able to actually identify how these proteins are co-occupying. For example, you can see here, these are a handful of different regulatory elements, which are like to be bound by proteins. And you can appreciate on these fibers labeled by 2 here, you can appreciate that you have actually occupancy at both this site 1, this site, as well as this site on some fibers, telling you that these proteins like to bind together, in a manner that sort of indicates that, this may be something essential for the function of this particular regulatory element. So what I'm trying to get across is that now we have a tool that really enables us to, get these, highly accurate sort of somatic variants and synchronously sort of identify the function. And also we can do this in a targeted manner, as I've showed you so far. But next we wanted to see is, does this actually work? Can we apply this to a situation where we do have a rare somatic variant? And using that situation, can we actually identify a specific functional impact of that variant? And so to accomplish this as part of the SMaHT network, we ended up taking this mixture of a melanoma cell line as well as a lymphoblastoid cell line, where we knew that there was a variant that was impacting the chromatin that only had a variant allele fraction of 1.4%. So this would be considered sort of a rare variant within that, and a rare somatic variant. And we've already shown that this specific variant actually disrupts the chromatin in this melanoma cell line. And specifically, it alters the occupancy of this protein called CTCF. And you can appreciate that here, in that you're not sort of seeing this sort of standard pattern of chromatin that you see on these top areas that do not have this specific variant. So when we applied targeted DAF-seq to this specific area, importantly, we were able to re-identify that this variant exists within this mixture and get a highly accurate quantification of the allele fraction of that variant, which means that we can accurately identify these somatic variants. And also, if we looked at those specific reads that contain this variant, we can appreciate that we're actually have similarly lost that chromatin architecture that is present on the reference, meaning that we can now co-measure both the variant itself as well as the functional impact of that variant in terms of chromatin architecture. So really what I'm emphasizing so far is that this methodology does appear to really address these major challenges that we have in the field in terms of trying to understand the functional impact of these somatic variants. In the last slide, I want to sort of talk about how we can not only do this in a targeted manner, but also do this in terms of measuring somatic impacts at individual cells. And specifically for this, what we ended up doing was taking individual cells and then treating it with this enzyme and then sorting it out into individual wells and now amplifying the whole genome of these single cells using this. And importantly, within a single cell, because humans are, we have 2 copies of every genome in the cell, we can actually measure how chromatin is on both of those copies of the genome within that single cell, because the pattern of how these stencils are happening on each genome are going to be different due to differences that we would anticipate that exist in terms of how the chromatin is organized on each of these 2 copies. So when we did this, we were able to actually demonstrate that we can actually have really good coverage of how the genome not only exists in terms of its genetic architecture, but also in terms of its structure from a chromatin perspective across nearly the entire human genome within a single cell, really a sort of unprecedented resolution in that regards. And furthermore, one of the sort of more surprising things when we did this methodology is we actually identified that outside of sort of the standard, what we would expect sort of genetic variation, there is a large amount of epigenetic or chromatin variation that exists between two individual cells, to the point that actually 60% of the regulatory elements within a given cell actually diverge in terms of their activity between two cells that are otherwise presumably identical, really meaning that we've sort of are able to now measure an unprecedented amount of somatic epimutations that we previously really have not been able to look at using prior technologies by leveraging the single cell DAF-seq. So really what I hope I've been able to emphasize and get across is, really some of the power that happens when you have these new technologies that enable you to look at somatic variation and not just measure the variants themselves, but really measure their functional impact within the cell. And this technology, I hope that, can really enable the SMaHT network to really not just uncover all the catalog of somatic variation across human tissues, but also uncover their functional impact. And there's many people I want to thank for this, including many of the people instrumental for the development of the DAF-seq methodology, such as Elliot Swanson and Easy Mao. And then many of the other people have been instrumental in terms of all the other aspects we've been doing here as part of SMaHT, both at our site as well as across the network. And thank you very much.